So let's continue our trip inside the muscle cell. We've already discovered many of the major parts of the muscle cell. And now we're going to concentrate on a part called the sarcomere. Sarco, again, means muscle, and mere means a small part of. So let's take a look at where this sarcomere is. So if you look in the upper left here, you can see part of a muscle cell. So this grayed out area is one part of a long skeletal muscle cell. They're telling us that it's a muscle cell because the covering of it is called the sarcolemma, which is the muscle cell membrane. We can see all these individual myofibrils inside the cell. So muscle cells are packed with the organelle myofibril, or long skinny pieces of something to do with muscle. These are about 5 or 6 myofibrils pulled out, and so they're pointing to one of those myofibrils. But here is that whole group of 1, 2, 3, 4, 5 myofibrils all pulled out next to each other. And we can see that the myofibril is covered with that blue network, which is called what? Can you find the word in this picture? It's a blue network called. Here it is over here, sarcoplasmic reticulum, or SR, sometimes we call it. That is the blue network. We can also see the end of each sarcoplasmic reticulum, which is called the terminal cistern. Next to this terminal cistern, we can see this white tube, which is called the what? That's right, it's called the T-tubule. The T stands for the word transverse. Transverse means going across. And so the T-tubule goes across the myofibril, not along the myofibril. So it's called the transverse tubule. And of course, the transverse tubule, we said, was going to allow electrical current to pass through the cell, and the sarcoplasmic reticulum is to store calcium ions. Now, what I'd like to bring your attention to is this very small part of a myofibril. So myofibrils are pretty much the length of a muscle cell. And this is a very teeny tiny part of the myofibril, and it's called a sarcomere. Sarco meaning muscle, and mere, as we said, meaning unit or small part of. And the end of the sarcomere is designated by this kind of wiggly line that is sometimes called a Z line, because it probably looks like the letter Z going back and forth. So we're going to take a closer look at this small part of a myofibril, and we know what's inside the myofibril. We said there's 2 types of myofilaments. Do you remember what they're called? Right, thick myofilaments, which are the red dots here, which are connected to these long red parts inside. Those are the thick myofilaments. And then the little blue dots, which represent the end of these smaller lines, which are called thin myofilaments. So let's take a look at them. So here we are, we've pulled off the sarcoplasmic reticulum, and now we can see one long myofibril, or at least part of it. And again, we can see the Z line, sometimes called the Z disc. It doesn't really matter, either one is correct. So there's one side of the sarcomere, and here's the other side. So this unit in here is a sarcomere, and of course there's other sarcomeres going down the myofibril. And if we expand that sarcomere so we can look at it a little bit more closely, this is what it would look like in 2 dimensions. So we have to imagine these lines in here, which are thick lines and thin lines, are now down here. And so this entire line of filaments is called the sarcomere. And here is one end of the sarcomere, the Z disc or the Z line over here. And then inside we can see some filaments. And this one with the little protrusions coming off of it is called the thick filament. It's red. And the blue one is called the thin filament. We can see that the thin filament is connected directly to the Z disc. So it's anchored right there on both sides. But it doesn't extend all the way across, it only extends part way from each side. The thick filament, on the other hand, goes all the way across the sarcomere. It's actually attached to another kind of molecule over here, kind of like clothesline attached to little anchors on each side. And it is that anchor that is attached to the Z disc, and the thick filament kind of hangs between them. So if we look at a sarcomere, we can see that there's a thin filament around a thick filament, a thin filament around a thick filament. Here's this thin filament and this one around this thick filament. And we can see that kind of the way it looks in here, thick filaments surrounded by thin filaments. But this is in 3D up here, and this is in 2D down here. So this is one sarcomere, one part of a myofibril. So if we review what we've learned so far, we can see that an entire muscle, which is attached to a bone, okay, so a muscle like the biceps muscle, covered by a covering called an epimysium, is divided into smaller units called fascicles. So each of these is a fascicle. And this is a fascicle, which is in turn a bundle of muscle cells. So there's many muscle cells in a fascicle. Here's one pulled out. Fascicles covered by the perimysium. If we look at one individual muscle cell, that muscle cell is covered by an endomysium. If we pulled something out of the muscle cell, it could be one of these myofibrils that we're pulling out. So there's one myofibril being pulled out. That's one of the organelles inside a muscle cell. And if we pulled things out of the myofibril, we could be pulling out the thin myofilaments and the thick myofilaments. They are the basically protein filaments that are making up the myofibril. So we can say that muscles are made of fascicles of muscle cells. Fascicles of muscle cells are made of muscle cells. Muscle cells are made up of myofibrils, and myofibrils are made up of myofilaments. So why don't you turn off the video for a minute and try to go through that so you understand the difference in organizational size of each different part of the muscle cell. Well, hopefully you do turn off the video and try it, because listening is not learning. You know from experience that when you watch a movie, you can't remember all the dialogue. You have to actually, if you're an actor, take that dialogue apart, think about it bit by bit, and learn it. And so all of these terms will be on your exam, and you have to know their relationship. So spend some time writing them down, talking about them out loud to yourself, teaching them to somebody else. They're all long, skinny things, so knowing the first letter is not going to help you either, because many of them begin with M. So you must learn all the terminology in this class for you to successfully pass the course. Okay, so we know the anatomy now. Let's start thinking about the actions of some of these parts. So since the myofibrils are able to shorten, and the muscle cell then shortens, and the muscle cell shortens, the entire muscle shortens, and when the entire muscle shortens, it contracts on a bone, causing movement. We have to understand how each of these things occurs. So we're going to be looking much more closely into the physiology or the function of all these different parts. So here's kind of my own cartoon drawing of a muscle cell. So if we imagine that this is a muscle cell, what's the covering of a muscle cell called? The actual, not the endomysium, but what the muscle cell is covered by? That's right, it's covered by the sarcolemma. That's the muscle cell membrane. And if this is a muscle cell, what are all these long, skinny organelles inside the muscle cell? That's right, these are myofibrils. And so the myofibrils are going to get smaller. That's what we mean by contraction. So the muscle cell is going to contract. It is going to get smaller. And that is due to the fact that the myofibrils inside them can get smaller. So the muscle cell will go from long to short because the myofibrils are going from long to short. So let's take a look at how the myofibrils do that. So now we're going to imagine again that this is a muscle cell, but we're only going to see 2 myofibrils within it. And this long structure is a cartoon version of a myofibril. And we said that myofibrils are made up of many sarcomeres. So sarcomeres are part of a myofibril. So each of these is a sarcomere. And this long structure is the organelle myofibril. Myofibrils are made up of sarcomeres. Kind of like a train is made up of many train cars. So the train would be the myofibril, and the train cars would be the sarcomeres. So if we zoomed in on them, we could see that each sarcomere looks like this, just like we saw in the past picture. There's the Z line on each end of the sarcomere, and then we can barely see some of the red thick filaments and the blue or purple thin filaments in here, but we can see kind of their construction. And now let's take a look when the cell shortens, right? So we shorten this cell from long to short. And now let's take a look at what happened to the sarcomeres. So take a look at these blue boxes. What's happened to the individual sarcomeres? That's right, they've shortened. And if we look at the actual, it's still a cartoon version of the sarcomere here, can you see what's happened? Can you see the Z lines here? And now you can see the Z lines here. Can you see that the distance between the Z lines is shortened? In other words, the sarcomere has shortened. It's gone from larger to smaller. And when the sarcomere shortens, the myofibril shortens. And when the myofibril shortens, the muscle cell shortens. So we're going to take a look at how these things happen in real life by taking a look at some cartoons here. So here is a cartoon version of the sarcomere. The Z lines are not really Z's, they're lines at the end of the cell here. So let me draw in those. We can see the Z lines here. And we can see the thick filaments, which are blue in this case, and the thin filaments, which are red. And I'd like you to notice that when the sarcomere shortens, the thin and thick filaments don't change shape. The Z lines are pushing, right, inwards. So the Z lines are coming in and shortening the entire sarcomere. So one more time, let's take a look at that. Notice the thick filaments are basically the same size. The thin filaments are the same size. It's just that the Z lines seem to be being pulled toward the center of the sarcomere. And that's what we want to take a look at here. How does that actually happen? So on this picture, we can see the Z line. And we can see what happens when the sarcomere shortens. Basically, the thin filaments are slid more towards the center, so that they're almost touching the center. And the entire width of the sarcomere shortens in. So if we have two adjacent sarcomeres and they each shorten, clearly the entire structure shortens. And we're going to try to take a look at what happens here, how that actually occurs. And we're going to concentrate on the microstructure and the interaction of the thin and thick filaments. So we can see the thick filaments have kind of little fingers sticking out that are actually going to contact the thin filaments. Let's take a look at how that would happen.